Hello, Gen Bio. Dr. Fry here with our second part of Lab 2, second part of Lab 1, atoms and molecules. So there are two basic things we want you to take from this segment of the lab, and that is we want you to be able to draw electron dot diagrams and build ball and stick models, okay? So an electron dot diagram looks something like this, okay? So in the top here, I have a chemical symbol for carbon, just the letter C, and then 4 dots around it. And each of those 4 dots is going to represent an electron in the outermost shell for carbon, okay? And we'll look at that more in depth as we progress through the presentation. The second thing we want you to be able to do is to build ball and stick models. So I have some built here to show you. This is a ball and stick model of carbon dioxide, where the black is representing carbon, the black ball, and it's bound to double bonds actually to 2 oxygens, carbon dioxide, okay? And there are a lot of other molecules and compounds we want you to be able to build, but just here's some basic ones. Here's 1 oxygen, 2 hydrogens, so oxygen is represented by the yellow, or the red. Hydrogen in this case is represented by the yellow. So we've got H2, H2O, okay, so water, okay. So these are our ball and stick models. So those are the 2 main parts of the lab today, of this portion. And before we progress further, I do want to just make sure that we're all on the same page as far as some of our basic definitions, so that we're all caught up as far as what the words that I'm using, the terminology, means. So first, an element is a substance that cannot be broken down to other substances by chemical reactions. You're probably by this point familiar with a periodic table of the elements. This lists all the elements and it makes up all of matter, okay? An atom, then, is the smallest unit of matter that retains the properties of each specific element, okay? And an atom is going to have a nucleus, so a center, and it's more like a space, not so much a distinct structure like the nucleus of a cell would be. It's more just a space at the center of the atom. The nucleus has a positively charged particle called a proton, a neutral charged particle called a neutron, and then outside of the nucleus in spheres, concentric rings, closer or further from the nucleus, there are electrons, which are negatively charged particles, which kind of occupy that space around the nucleus, okay? And valence electrons is our next definition here. Valence electrons are electrons in the atom's outermost ring or shell or orbital, we might say. So closest to the nucleus, you might have 1 ring that holds 2 electrons, and then outside of that, if there are more electrons associated with that element, then you might have another ring further out where more electrons occupy that space, okay? And so the number of electrons in the outermost shell will dictate how that atom is going to react with other atoms in its environment. And by react, I mean if it will form bonds, if it will be more likely to lose or gain electrons based on its electronegativity. So how it reacts with other atoms in the environment, okay? The valence electrons, those electrons in the outermost shell, they really dictate how that atom is going to react with others, okay? And again, by react, I mean forming bonds. A molecule then is formed when 2 or more atoms are held together by chemical bonds. So for instance, I have here a molecule of hydrogen. So I have 1 hydrogen, 2 hydrogens, and because they each have 1 electron, but their first shell is capable of holding 2 electrons, they tend to form a bond, molecular hydrogen. And they form this ratio, and they're more content, more stable in that state because they have complete valence shells. So that's why the valence electrons really dictate the behavior. So this is, say, molecular hydrogen. Molecular oxygen would be very similar. I think I have that one built as well for you. Molecular oxygen actually has a double bond then between two atoms of oxygen. The red is representing an oxygen atom. Okay. And then a compound is a substance that consists of two or more different elements. Basically, the difference here is two or more different elements that are combined in a fixed ratio. So for instance, H2O would be a compound. It's just two, it's got hydrogen and oxygen combined in a fixed ratio, 2 to 1. Okay, so that's a compound. Okay, pretty straightforward. Let's move on to our next slide then, which is a periodic table of the elements. You will find this in your lab manual, in your textbook, online. It's obviously here in the presentation. You can really access it anywhere, but you need to know your way around it, and you need to know what information you can find from a periodic table of the elements. So the first piece of information is you need to know, or you can find the chemical symbol, which is basically one or two letters that represents like shorthand for the element. So for instance, carbon is just the letter C. Hydrogen is just the letter H. Okay. Helium is going to be an H plus an E. Okay. And so forth and so on. That's the chemical symbol. The second thing you need to know, or you can find from your periodic table of the elements, is the atomic number. And the atomic number is always going to be equal to the number of protons for each element. Okay, and that number of protons is specific to each element. You cannot change the number of protons and have the same element. So if you look here, you've got hydrogen, this number 1, that's the atomic number. If you look at helium over here, number 2, that's the atomic number as well. So this is the number of protons. And then as you progress down and across our columns and rows, you just increase by 1. 3, 4, each of these elements then has an increasing number of protons. So if you were to change the number of protons, you wouldn't have the same element anymore. You'd have the next element on the periodic table. All right, that is different from the number of neutrons and the number of electrons. Those can vary for a specific element. So for instance, what you would normally have is an equal number of protons, neutrons, and electrons, kind of like baseline. Normal, you have the same number. So you would have 6 protons, which is our atomic number for carbon. And then you would have 6 electrons, and those are going to be occupying that space, the orbitals or shells outside of the nucleus. And then you more often than not have 6 neutrons also in the nucleus of that atom. However, I'll point out to you that our next term here, the atomic mass, is the number of protons plus the number of neutrons combined, okay? So as you can see, it's very close to 12 for carbon, 12.011, which you would understand then that there are 6 protons and just about 6 neutrons, because the atomic number is the combined number of protons and neutrons. But as you can see, the atomic mass is slightly above 12. It's 12.011, which is an average then. You should understand that it's an average of the number of protons plus the number of neutrons for all atoms of carbon existing in nature. What we can take from this then is that most atoms of carbon have 6 neutrons, because we know carbon always has 6 protons, and the atomic mass is really close to an even 12, right? But in smaller proportions, carbon can also exist with 7 as well as 8 neutrons, which make carbon 13 and carbon 14. Carbon can exist as carbon 12, carbon 13, or carbon 14, but most of it's going to be carbon 12 with 6 neutrons. Okay, and that's why this number, the atomic mass, is very close to 12, because that's the average of the number of neutrons in all of the atoms of carbon present in nature. Okay, all right, so let's move on to our next slide, which is very similar to the periodic table, but I find this picture very helpful when it comes to drawing your Lewis dot diagrams and thinking about how to how those electrons are oriented in space. So for instance, if we look at our top row here, we've got hydrogen and helium, same as we did on the periodic table we just looked at. But instead of having our chemical symbol and our atomic number and our atomic mass, we have a drawing basically of the nucleus and then the first orbital or shell that would have electrons in it. And since hydrogen is has the atomic number of 1, you're only going to have 1 electron to start with, baseline, and that's going to occupy the ring that's closest to the nucleus, okay? Since there's only 1, it's just hanging out there all by its lonesome, okay? Helium has 2. Now, I want to point out here that the first shell, the first orbital, it only holds 2 electrons. So this has a complete valence shell. And what that means then is that it's pretty stable. It's happy the way it is. It doesn't have a tendency to bond or give away or gain electrons. There's not a lot of reactivity. So anything that has a complete valence shell, which is characteristic of this final column, all of these, helium, neon, argon, and so forth, they have complete valence shells in this column. And so these are going to be very unreactive, or what we call inert elements. So they don't react very readily. This is completely opposite for those elements that have either just 1 electron in the valence shell or an almost complete valence shell, such as fluorine and chlorine. So I had said that the first shell, it holds 2 electrons. The second shell, if you look here, lithium, beryllium, boron, and so forth, over to neon, this second shell can hold up to 8 electrons. And so if it has all 8, it's going to be inert, unreactive, right? But if it has 7, it has an almost full valence shell. And so what it really wants to do is steal one more to complete that valence shell, because then it'll be stable and happy. So what tends to happen is that you've got a lot of reactivity between elements that are on the extremes. So oxygen, fluorine, sulfur, chlorine with lithium and sodium, because they have only 1 to gain or lose. And so you're probably familiar with, for instance, sodium chloride. Sodium chloride forms when this 1 lone electron in this third valence shell is taken away, basically stripped by chlorine, which wants to complete, it has a very strong electronegativity. It has a strong pull on that electron, and it wants to complete its valence shell. And so what happens is it usually steals that electron away from sodium. And what you have then is you've got the lack of an electron, the lack of a negatively charged particle here with sodium. So sodium then we say has a positive charge because it's lost that 1 electron, it's lost that negative charge. So it has a more positive charge associated with it. And chlorine then has taken that 1 electron to complete its almost full valence shell, and it will then have greater negative charge. It'll have gained an electron, it'll have more negative charge. And so we know that opposite and negative charges tend to attract to each other. And so what's going to happen is you're going to have an attraction between that negatively charged chlorine and the positively charged sodium, and they're going to be attracted and form table salt, sodium chloride, okay? So that's how that works. And I tell you all this to demonstrate how a lot of reactivity happens between the elements that have atoms with mostly empty or mostly full valence shells. The inner shells don't really have much to do with how the atom functions. It's really the valence shell, the outermost shell, that determines the chemical behavior then. Okay, so let's move on to our next slide and discuss a few more important concepts. I want to put a name to the concept that we just talked about. So valence. Valence is an atom's bonding capacity, and it usually equals the number of unpaired electrons that are required to complete the valence shell. So for instance, if you look at this picture of oxygen, which is just taken from the last slide, oxygen, you can see its atomic number is 8. There are 2 electrons in the innermost shell, and that shell is complete. And then the second shell, it's got 1, 2, 3, 4, 5, 6 electrons, and so it needs 2 more to be full, because that second shell, as well as the third shell, hold 8 possible electrons, okay? And it wants to get 8 in that outer shell. So it's going to want to be bonding with other things that can then share their electrons with oxygen to form bonds. So the valence of oxygen is going to be 2, because it's going to equal 2, because there are 2 unpaired electrons which are required to complete oxygen's outermost shell, or valence shell. Okay, so we'd say its valence is 2. Okay, all right, and then the next 3 terms, I'll start with an I, but don't get them mixed up. Okay, ions are atoms that have gained or lost an electron. We talked about this with sodium chloride, right? So those that have gained 1 or more electrons are going to have a negative charge. They are more negative because they got an extra electron. And we refer to those as anions. And then if they lost 1 or more electron, they're going to have a more positive charge associated with them. And we refer to those as cations. Okay, so it's going to be charged in one way or the other, an ion. Specifically, if it's a negatively charged ion, it's going to be an anion. If it's a positively charged ion, it's a cation. Okay. All right, we talked about that, like I said, with sodium and chloride. Sodium tends to give up its electron. It's going to have a positive charge, so it's going to be referred to as a cation. Whereas chlorine has gained that electron, is going to have a negative charge, and then it will be referred to as an anion. Okay. Okay, and then isotope, we discussed this too, but I want to put a name to it, is one of several different atomic forms of an element that have all the same number of protons, because that's specific to the element, but a different number of neutrons. So carbon, the example we talked about, exists in carbon-12, carbon-13, and carbon-14. These are called isotopes of carbon, okay, different numbers of neutrons, okay? And then an isomer, this is, and I'll continue this on the next slide as well. An isomer is a compound that has the same molecular formula as another compound. 2 compounds have the same molecular formula, but different physical structures, and therefore different properties. So structure often dictates function, how it reacts based on how it appears, how it's formed, right? And you can have compounds that have the exact same molecular formula, but they have different shapes. And we'll demonstrate this, like I said, more on the next slide, but I just want to direct your attention to this picture of butane, which has a molecular formula of C4, 1, 2, 3, 4 carbons, C4, H10. Okay, and you can see it written here, but that's indicating there are 4 carbons, C4, and 10 hydrogens, okay? And butane, looks just like this, with carbons all in a row and the hydrogens all attached to the limbs of the carbon. However, what you could have is you could have this 4th carbon maybe coming off of carbon-2 down below, and then have all the hydrogens around that, and that would have a very different structure and therefore a different function. And so that's an example of what an isomer is. So we have 3 different kinds of isomers that we expect you to know: enantiomers, structural isomers, which is what I just talked about, and then geometric isomers. Okay, so enantiomers are mirror images of each other. Structural isomers differ in the covalent arrangement of their bonds between their atoms. And then geometric isomers differ in arrangement around a double bond specifically. So let's look at our next slide so we can visualize that a little bit better. Okay, so the 3 different kinds of isomers we talked about are enantiomers, structural, and then geometric isomers, which are sometimes also called cis-trans isomers. Okay, So the first one is enantiomers. These are mirror images of each other. And I have built an example of this for you. You can also see it on the slide here. But you can see here that I have same number of atoms of specific elements. So again, like the black would be carbon, the red would be oxygen, the yellow would be hydrogen. So you've got, in each of these molecules, you've got the same molecular formula. But no matter which way I turn it, the left will never be the left. The left will never be the same, and the right will be the same at the same time, because they're mirror images of each other. You can see I can turn it multiple ways, but like if I have the blue facing towards the screen here, towards you, the green is going to be on the inside. The green is not always going to be on the left or the right, because again, they're mirror images. Okay, so that's what an enantiomer is, and you would see this in two different forms: the L and D form for levo and dextro, which are the Latin terms for left and right. Okay, so handedness, we often say like your hands would be an example of this, mirror images, right? Okay. All right, so that's enantiomers. That's one type of isomer. Number two here is structural isomers. Structural isomers, again, they have the same molecular formula, but different bonding partners. You can see here with butane, as I showed you on the last slide, and isobutane, they're both C4H10, but because that carbon 2 is bound to 3 different carbons instead of just 2, then the arrangement of those hydrogens changes, the whole structure changes, and so therefore it's going to have very different functionality associated with it. It's going to react differently in nature because it's shaped very differently. Okay. All right, and then the last type of isomer is geometric or cis-trans isomers, and they're shown here at the bottom, and they have a different arrangement of binding partners that's distinguished because of a double bond. So a double bond has a stabilizing effect on a compound. So for instance, I have here a double bond between 2 carbons, and I've got 2 different side groups here. So I've got hydrogens, and I think these are chlorines. It can be anything though. So you've got this double bond, which doesn't allow for rotation. Oopsie. It doesn't allow for rotation of the side groups. You can't ever have these 2 hydrogens be on opposite sides of that molecule. In this case, they are stabilized on the same side of the molecule. The hydrogens are on one side, the chlorines are on the other side, and they're stuck like that because of the solidity of that double bond. Just to show you by contrast, then I have a similar molecule, but because it's just a single bond, you can rotate that and you can have, say, the hydrogens on opposite sides, okay, of that molecule. Okay, but a cis-trans isomer, this would be one form where you have the same groups on one side, and on the other side, this would be the cis form. So if you look in our PowerPoint here, the cis isomer has the 2 similar groups on the same side and 2 hydrogens on the other side, versus the trans form would have that swapped. So you'd have your 2 hydrogens now on opposite sides and your 2 chlorines, or whatever it be, your X group, on opposite sides. So this would be the trans form of the isomer, and this would be the cis form of the isomer, okay, because they're the same cis. Okay. All right, so those are cis-trans geometric isomers. Let's move to our next slide then, where we discuss just a couple different types of bonds. So covalent bonds are most of the bonds we've been talking about so far, where these little sticks represent a sharing of a pair of valence electrons. So again, the valence electrons are those in the outermost shell, and they can be shared between 2 different atoms. So I'd shown you hydrogen, molecular hydrogen is H2. The yellow is representing hydrogen. It's just 2 sharing those. Each hydrogen donates 1 electron. They each had 1, right? and they're trying to have complete valence shells. They want to have access to 2 because that first shell holds 2 electrons. And so they're most happy, they're most stable when they have full valence shells. So they tend to then gravitate towards each other and form a bond. That's a covalent bond, okay? And we also, I've used the term, but I want to officially define electronegativity. Electronegativity is an atom's attraction for the electron in the covalent bond. Some atoms have a greater electronegativity. Like I said, the ones that have almost full valence shells, they have a much higher electronegativity than, say, hydrogen, which only has 1 lone electron. It's not going to hold on to that very tight, okay? So it's going to be more likely to give it away to something that has a higher electronegativity, like, say, oxygen, okay? All right, the next type of bond that, again, we've talked about, but I'll make sure we put a name to, is an ionic bond. An ionic bond is the attraction between 2 oppositely charged ions, so a cation and an anion. And we talked about this with sodium chloride. So what you have here is that sodium is going to be basically stripped of that electron. It's going to be stripped of it because of the high electronegativity of chlorine, and it's going to then have a negatively charged chloride ion and a positively charged sodium ion. And then that negative and positive, they're 2 opposite charges, they attract, okay? So it's a different type of bond. It's the attraction between the negative and the positively charged ion because one has already gained or lost their valence electrons, okay? All right. And then the last type of bond that we're going to talk about is a hydrogen bond. It's a type of comparatively weak chemical bond, but I don't want to belittle it because it's very influential in forming tertiary and quaternary structures of proteins, okay? It does help to determine the final structure of, say, a protein or other molecule. So a hydrogen bond, I don't want to belittle it, but it is less stable, I guess I would say. It's a slightly weaker chemical bond that's formed when there's a slight positive charge associated with a hydrogen atom, and it's attracted to a slightly negative atom of a polar covalent bond in another molecule. So there's a lot of words in there, so I'm going to direct you to the picture to demonstrate this concept. So hydrogen is going to more often than not have a slightly positive charge because, like I said, it only has that 1 electron negatively charged particle, right, in its valence shell. It's only got 1, and it's not going to have a very tight grip, so to speak, on that. So it's going to be more often than not getting stripped of that electron to something else, like oxygen, which has a stronger electronegativity, right? So when there's a shared relationship, such as with water, okay, so H2O, let me pull that out, I've got it somewhere here. When there is a bond between an oxygen and 2 hydrogens, because of the greater electronegativity of oxygen, that electron, the 1 electron that is associated with hydrogen, is going to be more closely pulled towards oxygen because of its greater electronegativity. So you're going to have a slightly positive charge associated with each of the hydrogens and a slightly negative charge associated with the oxygen, and that's demonstrated in the picture here as well. The red in the picture is oxygen. The white they're using instead of my yellow for the hydrogen, but because there's a greater electronegativity for oxygen, it's going to have that electron from hydrogen more often associated closer to the oxygen than towards the hydrogen. So it's going to have a slightly negative charge, and the hydrogens are going to have slightly positive charge. Okay, so then when you have more than 1 of those floating around, let me see if we can make another one quick, in space, you've got a slightly negative charge associated with this oxygen and a slightly positive charge associated with this hydrogen. And so that's going to attract. So you've got like multiple molecules. Now you could have more and more. I'm not going to build them all right now. You have more and more where there's a slight negative charge associated with the oxygen and a slight positive charge associated with the hydrogen. And these are going to form bonds called hydrogen bonds then, because of that positive negative attraction. Okay, and so between different molecules, okay, you're gonna have, and that's what causes the fluidity of water. You're gonna have these making and breaking of hydrogen bonds, which again allows for water to have a slight attraction to other molecules of water, but also to be able to be broken relatively easily. Okay, so I hope that makes sense. Any questions, obviously, you can ask me in lab or office hours as well. Okay. All right, so let's move to the next slide. I think we covered most of the content. Now I want to show you how to draw Lewis dot or electron dot diagrams. So for hydrogen or carbon, which are atoms, or anything else, oxygen, it's a very simple process to draw a Lewis dot diagram, okay? Basically, you need the chemical symbol, and you need to know how many electrons are in the valence shell of that element, okay? Those are the two pieces of information you need, and you can look at the periodic table for both of those, okay? So for instance, if we were drawing a Lewis dot diagram for hydrogen, we know the chemical symbol for that is just an H, right? And that's shown here. And that it only has one electron associated with it. So you really just put one dot next to that chemical symbol and there you go, done. Now, obviously that's the simplest example we're gonna get, and it's gonna get more complex from there. We can do it for atoms as well as for molecules and compounds, okay? So carbon is another common one. Carbon, if you look back at your periodic table, I'm gonna flip back to the periodic table, or better yet, to our electron distribution map, where you can see that carbon, we know it has 6 protons, and so we would baseline assume it's going to have 6 electrons. And we know that the first valence shell holds 2, and then that shell's complete. And then the remaining 4 electrons will have to be in the second shell or orbital. So this is what then that would look like. There's going to be 4 electrons in the valence shell. So I'm going to flip back to our current slide. Okay. So you would draw the chemical symbol for carbon, and then you would draw the 4 dots that represent each of the electrons in the valence shell for carbon. And you would start by putting one on each of the 4 sides of the chemical symbol: top, left, right, and bottom. Okay, top, bottom, left, right. You draw 1 electron on each of the 4 sides, and then if you were to have another electron that you needed to draw, then you go and put 2 on the top, 2 on the side, etc. So let's do that for oxygen. Let's say oxygen. Now I know, but I'm going to pull up the slide just to show you. If you look at oxygen, you've got 8 total protons, and so again, baseline, we're going to assume there are 8 electrons, 2 in the innermost shell, and then 1, 2, 3, 4, 5, 6 in the valence shell. So then what we would do then is we would draw our chemical symbol to start with, okay? And we would draw, so we're gonna do this: 1, 2, 3, 4, 5, 6, and that's all there is to it for oxygen, okay? And that's just for doing an electron dot diagram for an atom. Now, it gets a little bit more complicated when we want to do electron dot diagrams for molecules and compounds. So I'm going to erase my whiteboard here and get a little bit more room. So for instance, if we want to do water, we want to draw an electron dot diagram for water. We know that it's H2O. We need to consider how the electrons can be shared between our oxygen and our 2 hydrogens, so that the goal is that each atom has access to the number of electrons that will complete its valence shell. So for hydrogen, you're only talking about the first shell, so it really only needs access to 2. But for oxygen, we're into that second shell, if you recall. So it needs to have access to 8 electrons to be stable and happy and content that way. So let's go ahead and draw that as we would. You can see it on the PowerPoint, but I'm going to draw it as well. So you start with the chemical symbol. Well, I cannot do this backwards. Start with the chemical symbol, oxygen, and you got 2 hydrogens, right? Okay. Now oxygen, we know, we just looked at, has 6 electrons in its valence shell. So we're going to go ahead and go 1, 2, 3, 4, 5, 6. Okay, 6. And I'm going to change colors now for my hydrogens to make this more clear, because you've got oxygen with its 6, and then hydrogen. Each of the 2 hydrogens is going to donate 1 electron, okay, like so. And now what you can do to check yourself, if you want to just make sure that each of your elements, each of your atoms has access to a complete valence shell, what you can do is, I sometimes take another pencil or another color or something and draw a circle. Oh, my blue's not working. Draw a circle around, oh, we're just gonna have to use black. Around the chemical symbol and the number of electrons that it needs to complete its valence shell. So hydrogen only needs 2. So it's gonna be okay. Hydrogen, this hydrogen needs 2. It's gonna be stable and happy. Oxygen needs 8. And as you can see, you have that able to be met by making the 2 single covalent bonds with the 2 hydrogens. Okay, so it's got 1, 2, 3, 4, 5, 6, 7, 8. Okay, so they're shared then electrons. Okay, and if sometimes, if it makes it easier, sometimes what people do is just draw a line to represent, if you see that, sometimes they draw a line to represent the shared electrons. That's going to be 2 electrons, 1 from the hydrogen, 1 from the oxygen, 1 from the hydrogen, 1 from the oxygen. So you might see it drawn like that. Okay. All right. Now, again, that's still pretty simple. It gets a little bit more complex now when you consider that sometimes double bonds have to be used in order to allow for the valence shells to be complete. So for instance, the example we're going to look at now is carbon dioxide. Okay, so carbon, I'm just going to show you here, carbon has 4 electrons in its valence shell. Okay, so carbon, 1, 2, 3, 4. Okay, and then oxygen, we just established, has 6, right? So we could put our oxygens here. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. But when you go to check yourself and make sure that they each have complete valence shells, you're running into a little problem here. So if you look here, I try to draw my circle around carbon. It's got access to 1, 2, 3, 4, 5, 6. That's not a complete valence shell, and so that's not going to be stable or happy like that. And the oxygen as well. 1, 2, 3, 4, 5, 6, 7. Same on the other side, 7. That is not a complete valence shell. So that is not going to be the way that it's going to bond to make a fixed compound. So you need to consider that double bonds might be taking place here. Okay, so let's redraw this now using some double bonds and see if that fixes our problem. Okay, so we've got carbon, again, donating 4, right? So the way we draw this then is 1, 2, 3, 4 from the carbon. And then the oxygen each donates 6. And what you do to show that, I hope you're able to see this here, is that there's 1, 2, and then you kind of go catty corner, 3, 4, 5, 6 electrons donated by that oxygen. Same over here, okay? 1, 2, 3, 4, 5, 6. And this here, this is a double bond then. This and this, here and here, those are double bonds. But if I hadn't drawn the lines, you could see that now we can double check ourselves, right? And see that we've got complete valence shells because oxygen has access to 8, 1, 2, 3, 4, 5, 6, 7, 8 electrons, right? Carbon, again, will also have access to 8. I'll show you that clearly. Okay, so here's carbon. It has access to 8 electrons, and it's the same over here. Oxygen has access to 8. So those double bonds really allow for the valence shells to be complete between these different atoms of carbon and oxygen. Okay. All right. So any questions you can always ask me when we get to lab. Okay, if you're not clear on that. But we will be having you do some of those for practice in your lab manual, and make sure you get to know how to draw those electron dot diagrams. Okay. All right. Then, additionally, I've talked a lot about a few elements, carbon being one of them, because carbon has an unparalleled ability to form large, complex, diverse compounds. If you remember, our carbon, it had 4 electrons in its valence shell, so it can form, like, say, single bonds with hydrogens. So there could be 4 different hydrogens associated with that, and that makes methane, okay, CH4. Or it can form lots and lots of other bonds, like we just talked about with carbon dioxide. It can make double bonds with oxygen. There are tons and tons of ways, because it has 4 electrons, tons and tons of ways that it can combine to make larger and very diverse compounds. So some of these we should be familiar with, some of the carbon-based compounds. And we're going to put them into categories. So hydrocarbons are made up exclusively of carbon and hydrogen, just carbon and hydrogen. And the one I just drew, methane, is an example of that, okay? Carbohydrates then are made up of carbon and hydrogen and oxygen as well. And they often exist in the proportion CH2O. So 1 carbon to 2 hydrogens to 1 oxygen, okay? And glucose is a well-known carbohydrate. Okay, glucose, sugar, right, is a well-known carbohydrate that exists in this ratio. So glucose's molecular formula is C6H12O6. So again, it can be increments of that proportion, 1 carbon to 2 hydrogens to 1 oxygen, but it can exist in, they'll usually exist in that ratio in some manner. Okay. And then proteins are made up of long chains of amino acids which are bound together. So amino acids, we're going to look at those in a slide or two also. Amino acids they have a central carbon. That's called the alpha carbon. And then that's bound to 2 side groups that are in every single amino acid, okay? A carboxyl group and an amino group, okay? And then they've also got a hydrogen and an R group coming off of them. So again, these are the building blocks of all of our proteins, amino acids. They've got a carboxyl group on one side, an amino group on the other side, a hydrogen, and then an R group, which the R group can be a much larger, it's kind of like variable, okay? Lots of different options for that group. And then also carbon is present in DNA and lipids and lots of other macromolecules, okay? So in our model kits, we can demonstrate some of the smaller examples of these molecules, but they can get so much bigger that we can't, build them anymore because we've run out of balls and sticks. All right, But we traditionally have asked you to build these molecules in the lab to get a feel for how that works. I've been showing you some examples as we go through the presentation, but you should understand that the different colors represent different elements. So for instance, the carbon in my examples have always been black, and the hydrogens have always been yellow. In your kits in the lab, they might be white, etc. But each of those represents an atom of a specific element, and then the bonds are formed and they're indicated by the different wooden sticks or springs. Okay. All right. So let's just look at a couple more pictures of these, and then we'll wrap things up. So again, I said hydrocarbons, baseline, they're kind of simpler molecules just made up of hydrogen and carbon. And it can be any number of those. You could have, like we talked about, butene and isobutene. It was C4H10, right? That would be another example of a hydrocarbon, because all it has is hydrogen and carbon. But they can get very big, because you can have long, long chains of carbon surrounded by hydrogen, okay? Carbohydrates, then like glucose, you have carbon, hydrogen, and oxygen, okay? And that's shown here in number 2 picture here, okay? And then the third picture is showing a triglyceride, which has a fatty acid tail, which is a really long carbon hydrogen tail, a glycerol backbone, and that glycerol backbone is linked to the fatty acid tail by a ester linkage region, okay? The fourth picture here is showing you our amino acids that are binding together then to form a peptide chain or a protein, okay? So if you look here, I'd said that each of the amino acids has a carboxyl group, which we'll look at on the next slide, but this is the carboxyl group, and an amino group, okay? Carboxyl group has a carbon double bonded to an oxygen, and then an oxygen and a hydrogen coming off of it as well. And then an amino group is a nitrogen with 2 hydrogens coming off of it. And then the way that these amino acids are bound together is by what we call a dehydration reaction to form a peptide bond. And the reason we call it dehydration reaction is that you lose a water, dehydrate. So you take this amino group and this carboxyl group and remove an H2O through a reaction, and then that nitrogen and that carbon will be bound to one another, as you can see here, which is then a peptide bond, binding 2 amino acids together to form a chain, and that will ultimately form a protein, okay? And the last picture I have here is a nucleotide. So these are the bases for DNA and RNA, and that includes a phosphate group, a sugar, and a nitrogenous base. Now we can't expect you to build these, they get too big, but you, if given a picture, should be able to recognize that this is a nucleotide and it's the basis for DNA and RNA, okay? All right, last content slide is that we have 7 very important functional chemical groups, okay, that we want you to know what they look like, and we want you to know some properties associated with them and be able to build them. So for example, the first one is a hydroxyl group. You should know the name of it, hydroxyl group, and you should know that it is basically an oxygen and a hydrogen. So here I have a hydroxyl group right here. It's that simple, okay? Oxygen, hydrogen. You should be able to build that and know the properties associated with it, okay? Know what it is, be able to identify it. A carbonyl group, same thing. You should know that this is a carbon double bonded to an oxygen. I think I have that somewhere here too, but I seem to have lost it. A carbonyl group, carbon double bonded to an oxygen. And this can occur within a larger molecule, within like the backbone of a carbon string of carbons. You can have a double bonded oxygen coming off of that. And if it is within that backbone, we call it a ketone. If it's hanging off of the outside, like on the end of a larger molecule, then we call it an aldehyde. It's outside, it's an aldehyde. Inside, it's a ketone, okay? And then a carboxyl group, I had mentioned this as well as an amino group as being associated with our amino acids, which build our proteins, right? So you should know what they look like, okay, and know how they are used in building proteins, okay, like I just mentioned. And then sulfhydryl groups, a sulfur and a hydrogen. Phosphate group, again, you should know what that looks like, a phosphate and oxygens. And then the methyl group, which is 1 carbon, 3 hydrogens, and it would be attached to a larger molecule, okay? So review those, be able to tell me what they look like and the properties associated with them, okay? So that's really it for the presentations for lab 1. Okay, so at the end of this lab, we want you to be able to pipette, use the spectrophotometer, correctly format a standard curve figure, okay? Those were all covered in the first presentation. In this presentation, we talked about atoms and molecules. We want you to be able to draw electron diagrams and build structures using ball and stick models, okay? We do have post-lab work and our first quiz, which will be based on this material today and in the lab this week, okay? That'll be next week. All right, thank you very much. And if you have any questions, you can always contact me, well, in lab or in our office hours, okay? Thanks a lot. See you soon.